Welcome, and thank you for joining us today for the Automating Body Composition from Routinely Acquired CT Images Towards 3D Measurements webinar. My name is Vanessa Torres, and I will be your WebEx technical support host for today. Before we dive into today's meeting, I wanted to provide some quick information on the webinar platform. All lines have been muted on entry and will remain muted for the duration of the webinar. Please submit questions throughout the presentation using the chat or Q&A panel and select all panelists. A moderator will ask the comments and or questions on your behalf at the end of the presentation. If you require technical assistance, please feel free to contact me, the host, via the chat panel, and I'd be happy to assist you. If you need closed captioning, please refer to the link in the chat panel. We also wanted to let you know that today's webinar is being recorded. And now I'd like to turn it over to Dr. Joanne Elena to introduce our speaker.
Thank you, Vanessa. Our speaker today is Dr. Faisal Beg, who is a professor in the School of Engineering Science and the Faculty of Applied Sciences at Fraser University in Canada. He has a Ph.D. in biomedical engineering from Johns Hopkins University and has really been one of the leaders in automating the analysis of CT scans for body composition measures. We're very lucky to have him today to walk us through the latest advances and help us do this research cheaper and faster, or maybe just faster for more scans. But we're really lucky to get him today. And without further ado, I will turn it over to Dr. Beg. And I think, Vanessa, do we have, oh, great.
Okay. Thank you very much. Thank you, Joanne, for the kind introduction. And thank you and the organizers at NCI for the seminar series for inviting me to speak to you today about automating body composition from routinely acquired CT images towards 3D measurements. So my name is Mirza Faisal Beg. I'm a professor in the School of Engineering Science here at Fraser University in beautiful Vancouver, Canada. I come from this very nice place for hiking. And I'm going to talk to you about my experience in my work on automating the extraction of individual patient measures from CT images, leading to better prediction of clinical outcomes. Conflict disclosure, I'm the Chief Scientific Officer of Voronoi Health Analytics, a company that builds software for automating the measurement of body composition from CT images and medical images. So body composition is, as everyone knows, important for predicting clinical outcomes. From a simplifying point of view, the main question in the field is this. For the same clinical interventions, the clinical outcomes are different for different individuals. Why is that the case? Why is it that individuals undergoing the same surgery have different outcomes? Some have complications, others don't. Why is it that individuals who have chemotherapy, some have complications such as toxicity, severe toxicity, even death, and others don't? So numerous studies have shown that body composition, in particular lean skeletal muscle mass, is an important factor in predicting these clinical outcomes and explaining these clinical outcomes, such as toxicity, survival, infection, surgical complications, length of hospital stay, and so on. So we understand that the individual is the main variable here. Their body composition, their body habitus, is what defines one from the other. And if we had a magic wand and could measure the entire body composition, we can then associate some of these body composition measurements to certain clinical outcomes and predict which one of these measurements could lead to adverse clinical outcomes. So that's the importance of this field. Now, for the longest time, BMI, body mass index, and BSA, body surface area, have been the measurements of choice to use for body composition prediction. But for identical height and weight, that is the same BMI and BSA, skeletal muscle mass can vary substantially. This variation in the amount of skeletal muscle for individuals with the same BMI and BSA could explain the variability in clinical outcomes, such as toxicity from cancer treatment. CT images can provide accurate visualization of internal anatomy, the body structure, and are now routinely acquired in clinical workflows, and specifically in oncology. And the estimation of body composition measurements, such as lean skeletal muscle mass and fat, adipose tissue, from 2D single slice analysis at the lumbar third vertebra L3 or the thoracic fourth vertebra T4 is now widely used. On the right is shown over here two CT images at the L3 level showing that the muscle, which is in blue, is before and after chemotherapy is reduced after chemotherapy, and fat, which is in green, visceral fat and subcutaneous fat, are increased after chemotherapy. So we can actually see these changes in precise and gory detail. The three questions I'll be motivating and answering, hopefully answering in my talk today are, is 2D single slice based body composition analysis accurate enough for predicting individual clinical outcomes? Can this be improved? What are the challenges? Are there other measurements that can be extracted from CT images that can facilitate the discovery of additional risk factors and potentially provide targets for intervention? And a more practical question, how can these measurements be extracted with convenience to enable large-scale biomarker discovery research, as well as potential incorporation in individual treatment plans for precision medicine, the right dose, the right intervention, at the right time for the right individual? Now, body composition, forward, has come a long way from manual segmentation of sparsely sampled CT images, where it started in the 80s, to automatic measurements from high-resolution CT images. There exist several algorithms in literature now that have automated process of extracting muscle and fat measurements, typically from single slice at the L3 of the T4 levels. And now single slice analysis is basically the standard in the field. Now this has been there for about a decade, but the question to ask is 2D single slice based body composition analysis accurate enough for predicting individual clinical outcomes? So in this seminal paper from 2004, Shen et al. Showed that the same 2D slice may not be the best for both skeletal muscle and adipose tissue. In fact, for skeletal muscle, they showed that 5 centimeters above L4, L5 had the highest correlation between single slice measurements and whole body measurements. And for adipose tissue fat, 5 centimeters below L4, L5 had the highest correlation from single slice measurements to whole body measurements. The second point is that the regression prediction from regression lines based on this analysis may not be accurate for the individual. So let's look at the plot they showed in this very paper. And let's say that the individual's skeletal muscle area 5 centimeters above L4, L5 was measured to be 180 centimeters squared. Now the regression line will predict for that individual the whole body skeletal muscle volume of 32 liters. This is the middle horizontal line. But the individual could be anywhere between 28 liters, the lower horizontal line, and 40 liters, the upper horizontal line. So the prediction for the individual, if it's 28 liters actual skeletal muscle volume, is off by 4 liters. 4 divided by 28, 1/7, about 14%. If the individual is actually at 40, the error is about 8 liters. 8 divided by 40, 1/5, is 20%. So we have an error from 10 to 20% in the individual prediction from these regression equations. The same analysis works for adipose tissue as shown over here. Now the third point is that the error in single slice measurement, sometimes there's errors. So you're not really at the precise single slice measurement. That will further displace the vertical intercept with the regression equation and further add to errors. The fourth point is that these regression equations were actually measured on healthy individuals. So how do they generalize to individuals with sarcopenia, muscle loss, and sarcopenia due to different causes, different cancers, malnutrition, aging, and so on? And these were also individuals who were more or less younger individuals, 40 to 65 years of age. So how do these generalize to the elderly? So there are all these issues with respect to single slice analysis that need to be accounted for. Now, regression approach that was developed in this paper was actually developed for group studies. And I quote from this very paper, they say, because single slice imaging can only be used to distinguish reliably between subjects who differ in skeletal muscle volume by 10 to 12 liters and adipose tissue volume by 12 to 15 liters, the use of a single image skeletal muscle or adipose tissue area estimate, therefore, has limited application when individual subjects are evaluated. However, in group studies, using appropriate single slice area would require only 17% more subjects for estimating total body skeletal muscle and 6% more subjects for estimating total body adipose tissue than a multi-slice whole body volume protocol. So there was good reason to perform single slice analysis when manual segmentation was the only way available to estimate body composition. By increasing the number of subjects in the study by a little bit, 20%, one could save a lot of manual labor going from whole body volume analysis to a single slice analysis. However, with all these errors that creep in for individual outcome prediction, and the goal is precision medicine, therefore it is imperative that we try and reduce these errors wherever possible, as much as possible. So 3D volumetric measurements that are provided by the available CT images are therefore necessary, in my view, for improved prediction of individual clinical outcomes. So the current standard of single slice measurement that has been in place for the last 15 years, where we take a single slice, extract the 2D muscle area, extract 2D fat areas, and build body composition models, can hopefully now be replaced because of available technologies that automate the extraction of these measurements for the entire 3D imaging stack with 3D muscle volume and 3D fat volumes, leading to better body composition models, which then lead to better individual clinical predictions. The hypothesis is that 3D measurements of body composition may provide better prediction of individual clinical outcomes as compared to single slice-based measurements. Further, the signal for predicting clinical outcomes actually may be stronger in the 3D topography of distributions, the relative distribution of muscle, fat, and different organs at different levels of the body. And here I've shown some recent results from automatic segmentations of the entire CT stack. On the bottom row are automatic segmentation of skeletal muscle, and in comparison on the top are manual segmentation of the same skeletal muscle in the same individuals. And you can see there's a very good concordance between the two. Automatic algorithms do an amazing job despite the amazing variability that is seen in the individuals across the population. Now at L3 is somewhere over here in the horizontal line over here, and one can see that there will be some errors in predicting the whole body skeletal muscle from just one slice L3 cross-sectional area because of the variability up and down the body. Here are some segmentations of visceral fat and bone. This is fat inside the belly, below the skeletal muscle with respect to the bony structure. And again, you can see the amazing variability in visceral fat across the population. The bottom row is automatic segmentations provided by an algorithm. The top row are manual segmentations provided by tedious and very hard work by anatomists, carefully, painstakingly given each slice. And again, you see that if the goal is predicting the whole body visceral fat, using one slice at DL3 may or may not do that job justice. And now here is subcutaneous fat, which is fat outside the muscle and below the skin, or across these individuals. And again, the automatic segmentation on the bottom row agree very well with the manual segmentation in the top row. And I've also overlaid under this segmentation of subcutaneous fat, liver and spleen. And one can see the enormous variability in liver and spleen in these individuals. Now, the liver specifically is the site, is the center of hundreds of metabolic pathways. So it is not far-fetched to hypothesize that liver could be a very important contributor to clinical outcomes, but it's not currently analyzed in body composition workflows. Here are again some visualizations of liver, spleen, along with the lungs and the trachea, just to showcase these are all automatic results, by the way. And there are some errors as well. You can see this current model, which is a pre-production model. We're working very hard to refine these models in the background. They have some errors. They're not perfect as of now. And in fact, automated algorithms will never be perfect. I think there'll always be 10% errors in about 10% of the cases, which have to be dealt with manually. So I'll come back to that later on. But again, you see the amount of variability, enormous variability in the different structures in the body. The liver, the spleen, the different lungs, and even in the pericardial cavity in the mediastinum, you can see there's lots of variability. Some people have a large hole where the heart and the pericardial cavity is compared to others. Some people have larger lungs compared to others. All these measurements are, I think, important for predicting individual clinical outcomes, which should be taken into account. And here is shown the visceral fat overlaid on these internal organs. Again, you see this beautiful and very interesting variability of visceral fat with respect to the variability of different organs. Some individuals have larger livers but low visceral fat. Some individuals have smaller livers or average livers and low visceral fat, and smaller livers and high visceral fat. So I've again arranged this with respect to the amount of increasing visceral fat, and you can see from low visceral fat, the proportion of liver and spleen and lung and different organs in these individuals, showcasing the enormous variability we see in the population. And this variability could also underlie, to some extent, the variability seen in clinical outcomes. These bellies over here on the lower right are chock full of visceral fat. And the pericardial cavity in some individuals is really filled with visceral fat. And in some individuals, it's not as filled. And this would all be very important, I think, measurements, in my view, for predicting individual clinical outcomes. The other challenge for automatic segmentation algorithms, in spite of this, and in addition to this variability, is that sometimes very sick individuals have fluid edema that's present in their muscles. It's basically just fluid, plasma leaking out and accumulating in the muscle tissue or below the muscle tissue. So on the right over here is shown the raw CT image, and on the bottom of the paraspinal muscles is this fluid accumulation. Now, normal segmentation algorithms, which just rely on CT intensity, will capture this as skeletal muscle because it appears to be the same intensity as skeletal muscle, and therefore overestimates skeletal muscle mass for these individuals. However, we have trained our algorithms to be very careful and avoid this, and so far, it seems to be doing a very good job. Even the subcutaneous fat has these pocket holes in them that are fluid accumulation. For example, you can see this over here. And this is some kind of, granular fatty texture that comes apparent in these individuals who are really sick. And maybe this also predicts clinical outcomes. So now we have automatic tools for segmenting large databases, the entire CT stack of muscle, bone, and fat, and we're working very hard to also generate internal visceral organs and refine those, like the liver and the spleen, the kidney, and the pancreas, and all. And so this amazing set of measurements can now be used to develop better individual prediction models. So I want to now showcase how we will address one of the challenges, which is fields of view. Some individuals have CT images that are acquired for the thoracic region, some for the lumbar region. So over here is shown these measurements indexed by their vertebral levels. So at each vertebral level, we can go and measure the average volume or area in that vertebral level. So the skeletal muscle over here is shown in the red curve. It seems that the skeletal muscle increases in the shoulder pectoral area and then decreases. The lumbar third vertebra is somewhere over here. And the different tissues, subcutaneous fat, visceral fat, are shown in different colors. You can sort of see this one-dimensional signature curves that become the signature of the profiles of body composition for these individuals. And we can also now measure the tissue intensities, in HU tissue intensities, the texture of these tissues. So for example, over here, indexed again by the vertebral levels, subcutaneous fat in dark blue is over here. This is bone in dark blue is over here. Subcutaneous fat and visceral fat are over here, light blue and yellow. This is the lungs, the left and the right lung. This is the trachea. So these are air filled, so very low HU, minus 1,000, close to minus 1,000. And different individual organs are over here. Now we can use these to compare across different individuals. So for example, on the left is an individual who has very little fat, adipose tissue, but relatively more skeletal mass. So their skeletal mass curve is over here. An individual on the right, in comparison, has a huge amount of subcutaneous fat, and the subcutaneous fat chart is over here. So if you compare these two, the signatures of subcutaneous fat and skeletal muscle, the relative difference provides an interesting contrast and potentially could be very valuable for predicting their different responses to clinical interventions. And then we can also compare their tissue attenuations as a function of tissue, as a metric of tissue function. And so over here, this individual has a bone HU of around 400. And this individual has an average bone HU of 300. So what could be going on that explains this difference in attenuation factors for these different scans? Now, one thing that has to be remarked here is that these are not quantitative CTs, like they have not been calibrated so that the CT values, the HU values are equivalent. But still, these are interesting findings, and they sort of give us pause and thought of what could be going on that explains the change in the tissue intensities across these individuals. And now here's shown our population level volumetric tissue measurements by vertebral level. So we can combine these if we index them by vertebral level. So on the left is shown skeletal muscle over here. Then we have bone in blue, then the visceral adipose tissue, which is the belly fat, and the subcutaneous adipose tissue, which is the fat below the skin. And we can find some interesting patterns here. For example, in skeletal muscle, to my eyes, there seem to be two groups, a lower skeletal muscle group over here, and a higher skeletal muscle group over here in the thoracic region. Now, what explains these two subtypes? One with higher pectoral skeletal muscle and one with low pectoral skeletal muscle. And one could say, oh, it's maybe males and females. And I'll come back to that in the next slide. This is bone, and again, there's a lot of variability in bone intensity across these individuals. What explains this variability in bone intensity? Underlying health issues, osteoporosis maybe. The visceral abdominal tissue, belly fat, then lots of variability over here in different tissues, in different individuals, and then subcutaneous adipose fat as well. So now we can segregate these by different demographic criteria, for example, male and female. And it turns out that the hypothesis that females had lower skeletal muscle is not true. Actually, females and males, so females are in red dashed lines, and males are in blue solid lines. Both have roughly the same numbers of individuals in this high skeletal muscle group and sort of this lower skeletal muscle group over here. So there's something else explaining this distribution of skeletal muscle, maybe lifestyle. This is the distribution of bone, and again, both males and females show variation from low to high. And this is visceral adipose tissue. Again, there's no big patterns according to demographics, like sex here, male, female, the subcutaneous adipose tissues. But we can now showcase the enormous variability in these measurements in the whole CT image across the body as a result of these kinds of technologies. This is the attenuation in these tissues by vertebral level, the marker of function, so to speak. And there's not any distinct patterns here, but again, you can see the variability in tissue attenuation in skeletal muscle. Some individuals have very low skeletal muscle attenuation, some individuals have very high skeletal muscle attenuation. What's driving these differences? Bone is more or less more uniform, but still there's a lot of spread. Similarly, there's spread in visceral adipose tissue. What is driving the spread in this tissue type, in fat, for example, and subcutaneous adipose tissue? And these are really interesting questions revealed by this data. And again, we can segregate them by male and female and study them as to if there are any particular demographic differences and so on. So the second question that I started with was, what are potential other measurements extracted from CT images that can facilitate the discovery of additional biomarkers or risk factors of clinical outcomes? And I was motivating the previous slides that different organs like the liver and spleen, the lungs, the heart, the vascular system, they could be also very important in driving the individual response to clinical intervention. Further, biomarkers of tissue function, for example, texture, as a measure of fat infiltration or edema, fatty liver score, osteoporosis, bone health score, pericardial cavity fat. All of these could be very important in addressing individual predictions. So how do we discover which of these variables are important in predicting clinical outcomes? Well, machine learning is our recourse, and that will allow us to develop models that can predict individual clinical outcomes, not just identify group-based differences. So this is, as I see, the future of automated analysis, multiple organ network analysis from medical images using AI. A CT image is acquired, it's automatically segmented. Each of these organs, they can be measured in gory detail, the volumes, the tissue attenuations, along with socio-demographic factors, genetic factors, lifestyle, cardiometabolic factors. All of these measures, the extended body composition, so to speak, can be now used in AI and machine learning to build very precise models for prediction of individual clinical outcomes. And then this would help address and enable the goal of precision medicine for individualized treatment, the right dose, the right time for the right individual. And the hypothesis is that 3D measurements of multiple tissues may provide better prediction of individual clinical outcomes as compared to 3D measurements of skeletal muscle and adipose tissue alone, as compared to 2D single-slice based measurements. So what are the challenges? Well, I alluded to some of the challenges earlier. One of the challenges is if you start doing volumetric analysis, there's all of a sudden a huge amount of data. And quality controlling automatic segmentations, flagging poor segmentations, itself becomes a job. So it's really important to build good measures for quality control. I'll show you some of those measurements that we are trying to build to address this issue in a few slides. Standardization of field of view is important. Many of the retrospective studies are required for diagnostic purposes. And in those studies, the field of view is just basically what is of interest in diagnosis. Some are thoracic, some are lumbar, some are more extended fields of view. And if we have different fields of view, we can't compare apples and oranges. So maybe a standardization of fields of view could be a discussion that the community could have. Is that valuable? Is that important? If it reveals so much wealth of data for the individual body habitus, allowing us to make very precise predictions, would that be an adequate risk reward? In addition, we can also strive to develop models that relate partial fields of view to full fields of view, and improving on the single slice-based correlation analysis. Robustness to CT acquisition parameters. CTs are acquired in different clinical centers under different acquisition parameters, contrast, non-contrast, different voltages, and so on. And then they also have different artifacts in them. Being able to handle all of these gracefully is important. If you were to do automatic segmentation, being able to handle fluid retention that's sometimes present in edematous patients and not overestimate skeletal muscle as a result. And very important is we want to have better interfaces to enable quick editing of automatic segmentations that require editing, that require manual intervention. So my assertion is that 3D measurements should be as simple and even simpler to obtain than 2D measurements. If we can address these challenges, there are tremendous opportunities ahead. Improved, more accurate individual prediction models that incorporate a richer collection of predictive variables that allow us to discover new biomarkers of sarcopenia and obesity and particular subtypes of sarcopenia and obesity, and within individual longitudinal modeling of how the individual is changing and prediction of where their skeletal muscle and adipose tissue is going to be in the future as a result of clinical intervention will all be opportunities that can be enabled by these approaches. Now, how can these measurements be extracted with convenience? And this is a practical issue, because it's all good and fine to have this amazing theoretical framework, but in order to incorporate these in clinical workflows, they need to be easy to do, easy to perform. Even to perform large-scale research studies, it cannot be done if there are no good tools. So we've had a history of, in my lab, to try and build and share tools and technologies and translate them out to the community for research purposes. We started with ABAX as a cytomatic plugin, and numerous research groups around the world are using this, and we're thankful for them for trying this out and for supporting us. Then we started with the data analysis facilitation suite, and what this enabled was to do batch processing. So for cytomatic plugin, you have to load one image, and you can only analyze one image at a time. If you're trying to do 100,000 images, good luck with that. So the data analysis facilitation suite, you point this tool to the input folder that contains your 100,000 images, an output folder where you want to store the results, hit run. It does everything in a batch processing mode, and go have your coffee, overnight, come back next morning, you have the results. Now, we recently released the DAPS 2.0, which further adds a lot of feedback from users that led us to these very different tools in it. So here again, we have addressed the needs of users and provided some additional tools like curating DICOMs. I'm going to show that in the next slide. And we recently released DAPS 2.05, which allow you to do a very easy and efficient marking of L3 and T4 levels with our very customized interface. So our whole goal has been to not just perform the research, but also to deliver these tools in a form that can be used for large-scale 3D volumetric body composition studies. And the two main pillars of our approach has been this DAPS, the data analysis and classification suite, which is an AI automation powerhouse, and then the CT annotation and segmentation tool, which is the manual intervention interface, where some editing that is required can be done efficiently and effortlessly. So a lot of thinking has gone into designing simple, intuitive, minimalist interfaces. So our interfaces will only provide the tools and the options necessary to perform this work and no more. Very simple. So here's the DAS interface on the left, and here's the annotator and the segmenter interfaces on the right. So DAS, what are some of the key features of DAS? So one of the key features is DICOM curation. For those who work in research in this area, know that when you download data from PACS, the Picture Archival Communication Systems, the data can come up with a large jumble of lots of files. Individual axial CTs are mixed up in this jumble, and separating them into individual folders is itself a huge challenge. With DAS, you point the input folder to this working folder where the data has been downloaded, and it works on curating this, and it organizes them into individual specific folders where all the individual Excel files are been carefully organized. So you can go from a PACS data dump to 100% organized analysis-ready data in a matter of a few minutes. You can also anonymize your DICOMs, and it gives you the option to preserve certain fields that you want to preserve or force remove certain fields when you want to force remove. And further, it never compromises original data, but always making a copy of the data when writing out the disk. So here's the segmentation module. So now we can do 3D segmentation. You point the DAS module to the working folder where you have curated your scans, and in no time, you can have 3D segmentation ready and accomplish population-based volumetric body composition analysis in just a few hours. Let me play that again. So this is very nice. Again, a minimalist interface. We worked very hard to define and design, and now it's available. We've also worked hard to provide easy and intuitive quality control of these segmentations by making an interface that provides all the segmentations as these thumbnails in one PDF document. And then you can get the PDF document for the entire database you want to analyze in a convenient folder, and you can just flip through them. So you can see the segmentation and the raw scans side by side, which enables you to flag poor quality segmentations. Now, those segmentations that may require editing, we have the Multiview navigation segmenter application that allows you to address the needs for editing. So there's some jitter in the video. Play it again. Okay. So here again, you point the segmenter app to the working directory where the segmentation has been stored. And instead of going one by one, it loads the entire set of folders in your working directory into its internal database. And now you do not have to go and load individual images and their segmentations one by one. So you can scroll through each of these folders, each of these individuals through this app itself by using previous and next. And then you load, you get a view like this over here, and then it allows you to segment and edit these tissue types that are of interest. There are two main tools here. There's a pen tool to add segmentations and erase tool to remove segmentations. And certain presets allow you to more interactively and finely control segmentation. So here's an example of this pen tool being used to mark a segmentation, and there's a lot of shortcuts that allow you to go across slices very fast. So it makes it very easy to address this need as well. The AVA, the Automatic Vertebral Annotation Engine, is the annotator tool. And now if you want DAS to annotate your vertebral landmarks, while you're running DAS, you select the annotation box. And if you're only interested in L3 study, so these are all options that we've kept to be backward compatible, then you type L3 in the box over here, and the DAS engine will go and try and identify the L3 slices for you. If you're interested in T4, type T4, and you try and identify the T4 slices for you. If you're interested in all of volumetric analysis, the DAS engine will go and identify all the vertebral levels from T1 to the sacrum pelvic spine. Now, if they need to be edited, you can go in the annotator app and edit the automatically placed slice landmarks, vertebral landmarks. Or if you want, you can actually do a manual analysis by not selecting annotate and letting you or a technician go and put the landmarks of L3, T4, and all the other vertebral levels that you may be interested in. Now, this is the main difficulty, a challenge, is how do we address different fields of view that have been retrospectively acquired. because now you cannot compare skeletal muscle and fat from one field of view to another field of view. So here there are three fields of views. And if we just do a volumetric analysis, that's a challenge. There will be larger skeletal muscle in one individual compared to another just because of larger fields of view. But with this vertebral level landmarking, and especially with this annotator tool, now we can extract individual vertebral levels and compare skeletal compare body composition measurements across these vertebral levels. So in summary, we have this automatic organization and 3D quantification with data analysis specification suite DAS, allows us to do data configuration, like anonymization, ABAX volumetric segmentations, vertebral annotations, and then finally provides us with tissue statistics in a CSV file that can be loaded in Excel, where for every slice, all these body composition measurements are noted along with tissue intensities. The slices are also identified according to the vertebral levels they belong to enable you to do a vertebral level by vertebral level analysis. So here is this high quality throughput workflow with CT annotation segmentation tool, load the images that have been automatically segmented into the CAT tools, edit those images, save, and repeat. So it makes it really efficient to provide this long, large database analysis. So we are working very hard in the background to perfect our tools, iron out the bugs. It's almost ready. It'll be coming to Windows very soon, next year. And so more details can be found at this link. So in summary, I started with motivating 3 questions. Is 2D single slice body composition analysis accurate enough? And I hope I have motivated the need to go to 3D analysis, not only because of better correlation, but because the volume of information that 3D analysis provides is vastly greater and richer than 2D slides can ever provide. The different visceral organs, the lung, the heart, all of these are really probably very important in predicting individual outcomes. What are the measurements that can be extracted? So the measurements are the other organs that can and should be analyzed in these workflows. And how can these be extracted with convenience? So there's a tool that we have built now that can be used to provide to do this analysis. So our DAPS tool allows us to take individual CT images, segment them. These become the x-axis, the variables that are driving the clinical outcomes. And based on these, the next big step is the prediction models, building these with machine learning from large databases. Now, this research needs to be done in lots of different domains, in different diseases, different cancers. And then once these models have been built, this will be the next big step, that when a new patient comes in, that patient's image gets segmented, and the clinical outcomes are predicted with better precision, better accuracy than ever possible before. So I've shared with you all my hypotheses and thinking, and if you like it, please take these and build your grants around it. I'll be happy to collaborate with you if that's suitable. We can provide you the segmentations and also the expertise and machine learning models to build these clinical outcome prediction models for the individuals. So I just wanted to thank here and acknowledge the amazing team that I've worked with, both at Fraser University in my lab and at Warnock Health Analytics, where we have a talented team of AI engineers, anatomists, software developers, working very hard behind the scenes to develop high-quality automation tools. My collaborators, all the research participants who provide images and publicly accessible databases, such as TCIA that facilitate this research. The audience, for taking an hour of your time, a valuable time, and attending this talk. I hope I was able to create some value for you. Funding agencies, National Science Engineering Research Council, NSERC, CHR, Canadian Institute of Health Research, and also the NIH for funding parts of this research. Michael Smith Foundation for Health Research as well. And thank you, everyone. I'll be happy to take questions.
Thank you so much, Faisal. This is wonderful. And you mentioned it might be provocative, and it certainly, I think, has led to a bunch of questions. So I'll get started. Lots of compliments and lots of comments on how great your images are. Are there data on the use of manual or automatic segmentation for pediatric populations?
Not as of now. This is all very recent work. We are thinking about it, want to do it. we'll be happy to collaborate with you if you have data that we can segment and provide to you. Yeah, so it's a work in progress, absolutely, but it needs to be done.
Great. Let's see. Wondering about the correlations between manual and automated segmentation.
Right, so that's again a good question. So with this large volume of data, how do you generate manual segmentations? Because it's very tedious to generate manual segmentations. So the way is to generate automatic segmentations and then correct them and generate manual segmentations. So we are working on that. So again, these are studies that need to be taken into in different disciplines, different domains, under different scanning conditions, and a lot of validation needs to be done in different scenarios before these can be ready for prime time individual clinical prediction. So again, we are happy to collaborate with different groups who are interested in validating these measurements in their own populations.
Great. What's the sensitivity of the technique, and can you detect nerve bundles, blood vessels?
Blood vessels, yes. Nerve bundles, I'm not sure. I don't know. We have to work on that.
Sounds challenging. For the vertebrae level measurement plots, did you say that you could predict other parts of the torso using algorithm based only if you have thoracic or lumbar scans? So
interestingly, yes, the algorithm does seem to work quite well on other parts of the body that is not seen. And that's surprising. That's very interesting. But again, we need to have proper data and proper studies to establish that.
Do you need a whole body scan to generate these images?
No, these are generated from just the fields of view that we have right now. These are not whole body, but they are good enough for a proof of concept start. And I think this shows that this is feasible now. And if we have whole body scans, we'd be very happy to generalize these measurements to whole body. But we don't have whole body scans. So if anybody has whole body scans, would like to collaborate with us, we'd be very happy to hear it from you.
A related question was, what sort of burden is on the participant? How long does it take on the participant's side?
So CT is amazing in this respect because a CT scan is just a few seconds, maybe 1, maybe 2, maybe 3 seconds. So the time burden to go from a lumbar scan to maybe a shoulder to hip scan is 1 or 2 seconds extra, 3 seconds extra. MRI, the scanning time is much longer, several minutes to 10 minutes, and depending on the field of view, that can get extended quite a bit. So for CT, this is an opportunity for acquiring a larger field of view if it is feasible.
For someone who asks participants to do food frequency questionnaires, a few seconds extra time is nothing at all. What is the metric that you provide? Attenuation or AT10 and volume per vertebrae, or labeled voxel?
Well, we can provide all the measurements that you need. So you can provide each of these. We can provide the number of voxels. You can now take the measurement of each voxel, convert those into centimeter cubed. You can provide single slice area measurements. We can provide single slice and thickness-based volume measurements. It all depends on your user needs. Once we have segmented these scans, generating these measurements is no extra cost. Very trivial.
Great. We have a couple more questions. I'll bundle them together about cost. Considering the man hours used in manual reconstructions, is automated technique cheaper and or faster? If it is, can people use it to look at their gain in muscle or loss in fat? And related, what is the difference in cost for 2D versus 3D?
Okay, so in terms of cost, the main cost in 3D is the amount of time you need to segment these scans. But hopefully, I've motivated that with automatic tools, that cost is substantially reduced. So hopefully, this will unleash a large number of 3D-based studies that will look at these measurements. I'm not sure what else was the question. Can you please repeat, Joanne?
Yes. So I think you got, oh, the other question is, do you think that this would be, do you ever think it'll be cheap enough that we could use it practically to look at gains in muscle mass or loss in fat?
Absolutely.
So,
Absolutely.
Yeah.
I think the benefits of using these measurements will be so amazing that, and the cost will be relatively so cheap that it will be definitely very practical and very feasible and very exciting to use these measurements and various clinical studies and individual clinical workflows as well. That's the next big step.
Great. Can your tool automatically segment MRI scans?
No, this is only for CT for now. We are working on MRI scans in the background.
Okay. What are the accuracies of the tools you have shown, and are there any publications? You mentioned some of this, but
yeah. So the 3D tool is, state of art. It's just hot off the press. So there are no publications yet. So I'm going to write results up relatively soon and submit them for publication. In terms of the 2D analysis, our 2D methods have been very heavily validated in lots and lots of publications. Those publications are available on the website.
Can this tool measure intramuscular adipose tissue?
Yes, absolutely. It can measure intramuscular adipose tissue.
Oh, this is a nice one. I have no questions, but want to thank you for your presentation. As I've segmented hundreds of 2D slices manually myself, I enjoyed it greatly. One can hear the passion for your topic, and it's amazing. Always nice to hear nice things, too. I agree. Is the algorithm sensitive to make, to the type of CT machine that acquires the image?
So far, no. We have trained the algorithm and tested it on a wide variety of images from a wide variety of CT scanners, and it seems to work really well in all of those conditions. So even on the slide over here, these images are all taken from different CT scanners, and it seems to be reasonably robust to those variations. But again, large scale studies need to be done. So the main test of the tool is when it's applied in the field for independently for images that were never seen on for validation on independent test images. So once those are available and users start using our tool, we'll be definitely interested in seeing the results. And my hope is that this is quite robust to different CT scanners and different acquisition variables.
Great. This might be, this is a great question, but it might be beyond the scope of your presentation, but how can body composition data be used to individualize chemotherapy or therapeutic agents? Do you want to take that, or should we just leave it as a very important question?
Well, I can give a, according to my understanding, by me, no means I'm an expert in chemotherapy. So in chemotherapy, the dose is decided based on BMI, body mass index, or BSA, body surface area, or sometimes it's flat dosing. Everybody gets the same dose. Now, as I understand it, for individuals that have a large amount of skeletal muscle mass, the same dose would be an underdose. And for individuals that have a low amount of skeletal muscle mass, the same dose could be an overdose. So based on BMI, you select a dose, but the dose will or could become an underdose if the individual has high skeletal muscle mass, or an overdose if the individual has low skeletal muscle mass. So BMI and BSA are not good variables to predict the prevalence or the presence of skeletal muscle in the individual. And they're certainly not good enough to predict the richness of the other body composition, the extended body composition variables in that individual. So therefore, BMI and BSA dosing for chemotherapy could be leading to adverse clinical outcomes and could be improved by the availability of these richer measurements.
Great. Several comments asking you to publish this soon and asking that you put citations available on your on your Veroni Health website.
Mm-hmm.
Also, questions if the software for 3D will be available for purchase.
Yes, it will be available for purchase.
Okay. Okay. Let's see. How do you envision body composition analysis from CT interacting with tumor radiomics, extraction of textural features from tumor CTs that could predict cancer outcomes?
Great question. So tumor radiomics tries to predict the tumor intensity values, and based on intensity values, some aspect of function of the tumor. I think this is very complementary, but we can also measure the additional organs around the tumor, the skeletal muscle mass, the fat, the bone. All of these form the extended body composition, body habitus, and they may also influence how the tumor responds in that environment. So I think it's all very complementary. By making very precise and very rich measurements, entire goal of precision medicine is becoming more and more enabled.
Great. Can the analysis be done from top of the head to the bottom of the toes, and can bones be identified and volume determined?
Yes, we can do the analysis top of the head, the bottom of the toe. For the identification of the bones, we only have so far a way of identifying vertebral labels, the slices belonging to each vertebral label. So the further work is going on to identify different bones and labeling different bones. And then we are hoping to provide individual bone-based measurements that can lead to people studying various aspects of bone health and variability in the individual.
Does your group train people to use your software, or will you offer something like that?
Yes, we offer a full service package as well. If individuals want, you can just give us images. We perform all the measurements and get back those measurements in a convenient CSV file. Or we also provide training to individuals and groups who may be interested in using these softwares.
Great. How far away is the automated 3D segmentation of individual organs or quantification of, say, liver fat?
So we have a working prototype. We are refining these. So we are not ready to release them just yet. We want to refine them further. So I would say between 3 to 6 months is when we will have the visceral organs and their health scores as well.
Great. How do you, if you'll think a bit about how to use this in clinical practice, how do you expect the use of information of multiple organs and tissue would be used?
So as I envision it, when an individual gets scanned in a CT scanner, currently the CT image is used only for diagnosis. Now a lot of studies have shown that we can actually make use of the body composition information in that CT image to design particular treatments, to tailor those treatments, design the dose of chemotherapy medicines, and also actually predict what are the chances of the individual to have what we call adverse clinical outcomes. So if an individual with a certain body composition type is at risk for maybe surgical complications, maybe the surgery could be avoided and something else substituted in its place. If the individual is at risk for chemotoxicity, maybe the dose can be reduced or tailored or given more frequently or things like that. So I think all this, as I foresee it, is going to be a norm, a routine in the future where these images will be acquired and not just used for diagnosis, but also used to very precisely estimate the individual's own body habitus, their body structure and function, different organs, and all of these. And then the work that still needs to be done is to identify which of these organs, which of these measurements are important, and what constellation of measurements lead to different clinical outcomes, so that when a new individual comes in, we can very precisely predict the response that they will generate if we were given a clinical intervention. So that's the goal.
So exciting. Okay. Talking about fluid accumulation, do you think that this would work for peritoneum for ascites?
I'm not sure what.
Okay. Fluid and yeah.
So I can indicate that that's fine.
Okay. Are you currently using only CT images for automated segmentation, or can you also set? Oh, we already mentioned that you can't use MRI.
Yes. Not yet. Yes.
Yeah.
Okay.
Would your algorithm work better with dual energy CT images?
I'm not sure if it works better, but we'd be happy to give it a try.
Okay. Another request for training. I think you might be busy after this talk.
Okay.
Nice presentation. Again, many compliments. I'm skipping most of them. Does the 3D measurement accurately detect skeletal muscle fat infiltration? Oh, you mentioned that already, but the answer is yes. Is there a minimum dose to get good segmentation? Could low dose scan modes also be used?
Again, that's a good question. These are all standard clinical scans that are available in publicly accessible databases. We just use them without really looking at a particular dose of the CT. So if the database included low-dose CT scans, then we probably have good results. If they didn't, then that'll be something new. Again, we don't have data. If somebody has data that we can work on, we'd be happy to give it a try.
Okay. Are you estimating presence of IV contrast and phase of contrast to adjust tissue attenuation?
No, not now. And that's what I alluded to in my talk, is that these are quantitative CT. They have not been calibrated so that the bone and the muscle and the fat are all sort of standard. So there's still some work to be done to quantitatively calibrate these so that the HU values can be compared across different studies. Now, in a study, if all individuals are acquired with the same CT scanner, and if the CT scanner is calibrated and so on, so then they can be compared. But as it stands, that is a variable of interest that has to be dealt with carefully.
Great. Can the software be applied to the Cancer Imaging Archive, which is a large archive of medical images of cancer accessible for public download?
Absolutely. Absolutely. We have tested on TCLE images, and it works quite well.
Great. Oh, you just answered the question about detecting the use of contrast material. A second question. This one we've got. Can your program estimate the tissues that are cut off on the CT? If the patient's too large to fit on the scanner.
No, this is a very direct segmentation algorithm. It only measures what's available in the CT image. Of course, the hope is that we will build algorithms, models that will take smaller fields of view to larger fields of view. That's, again, work in progress. Has to be done.
Great. Will the 3D skeletal muscle segmentation avoid major vasculature since the attenuation between the two tissue types are very similar?
Right. It all depends on what is the template for each organ. So, for example, right now, liver, we basically segment the outline of the liver, and everything inside the outline is a part of the liver. So it would also include some aspect of blood vessels. Now, we work hard to avoid major blood vessels, but the smaller blood vessels that are in the center of the human eye are probably part of the liver tissue. It all depends on it, and we are hoping that as time goes on, we refine these models more and more and more to avoid major blood vessels, very, very small blood vessels, identify those, label those, so that in the future we can provide a very finely segmented and labeled CT scan with all the small to large features.
Great. And the last question, you may have answered. Does 3D segmentation work on both contrast and non-contrast CT scans?
Yes, it does. So in our database that we've been applying and training on, we included both contrast and non-contrast scans, and it handles both of them fully gracefully.
Wonderful. Well, you finished right on time, 4 o'clock to the dot. Thank you. I guess I should not expect nothing less from an engineer. Thank you. And so thank you to everyone joining us today. Our next speaker will be in the new year. Hopefully 2021 is a lot better than 2020. and we will have Dr. Helena Furberg talking about the BMI paradox in kidney cancer. We have a website already up and running for registration. Unfortunately, you do have to register each month for these talks, but I am going to put the registration link into the chat box right now for anybody who wishes to register right now. Otherwise, expect an email from me in a few weeks. Have a wonderful holiday season, and I hope to see you all in 2021. And thank you again, Dr. Beg. This is a great, great talk. Thank you.
Thank you for inviting me. Goodbye.
Bye. Registration.